of BIO110's 3 student learning objectives. You can find these in the syllabus. 2 of them directly concern the ability of students to connect experimentation with conclusions as a pathway to building scientific knowledge. It's not that we expect every BIO110 student to become career scientific researchers. Some of you will, but this is not a class to train students for academic research careers. Every student, however, needs to have the same basic tools of inductive and deductive logic that are employed by professional scientists. Given several pieces of information, what can be legitimately concluded, and maybe even more importantly, what kinds of things would not be unequivocally supported by the information provided? What is it about certain results that constitute unequivocal support for a given conclusion? This is science. It's also what thinking citizens do as they process the flow of information that comes into their awareness through the media as well as by direct observation. Now, the opposite of processing information and reaching defensible and well thought out conclusions would be to accept statements without requiring validation by facts. This is non-science, and it involves no thinking at all. A demonstrably false statement is not made any more valid if it's repeated loudly and often. In fact, this is the opposite of science, and to the extent that our knowledge of the true nature of the universe is a good thing in the sense that it informs and speeds our progress toward greater well-being or some other generally agreed upon goal, that would mean logically that it's a truly bad thing for people to accept unsupported claims. Thinking like a scientist is thus a worthy aspiration for every person, period. It's not the easiest thing you'll ever do. It requires discipline and effort, and having a bit of training doesn't hurt either. And here's where this lab comes in. As in the previous lab's video, there will be points here where the playback will pause for you to answer questions, and this will constitute your pre-lab quiz. Some of the questions will be drawn from the information in the handout, not necessarily content that's spoken or shown in this video. You should read through the handout before moving onward with this video. The need for discipline might have been evident as you read through the introduction. How are an observation of a phenomenon, a question asked about the phenomenon, and a hypothesis to explain the phenomenon, how are these 3 things both related and different? One person may find the distinction between these things to be pointless attention to detail, while another person considers this to be discipline. And for understanding the scientific method, you're better off erring toward the latter. Differences that might seem trivial when viewed casually actually turn out to be salient when there's a need for greater rigor. And this is an example of that extra effort required in order to think like a scientist. You'll have the opportunity to practice your skills in this area at various points in our lab exercise. But more than what you're doing in this class this week, you might be practicing these skills on your own, based on the things you encounter within your own orbit. What is the observation? What is the question? What is the hypothesized explanation for the phenomenon? Is the hypothesis testable? What kind of experiment would be useful for evaluating the validity of the hypothesis? What kind of result is needed to support the hypothesis? And what kind of result would clearly refute the hypothesis? Can you describe an experiment with manipulated variables that would be able to support the hypothesis with some results and refute the hypothesis with different results? What kinds of confounding variables might be problematic for your experimental design, and how might those confounding variables be controlled? The main focus of this lab is to develop your ability to address all of the questions I just listed, but really the task at hand requires nothing more than what you carry around with you at all times. You certainly don't need to restrict this kind of thinking to just when you're in a science lab. This is an exercise that you should continue on your own time, outside of class, for the remainder of the semester and the remainder of your life. But for now, your immediate goal is to be able to take any real-world phenomenon that your instructor gives you and identify a potential explanation, and this would be your hypothesis. For instance, I could tell you that the 1,000 young fish put into a lake were all the same size initially, but they've been growing at different rates. This would be the observation. You should be able to come up with a hypothesized explanation for the observation, such as the fish are getting unequal amounts of food because some are more aggressive than others, or the two sexes of the fish grow at different rates, or you can come up with your own idea here, right? And then you should be able to follow that up with an experimental protocol for evaluating your hypothesis, delineating what results would support the hypothesis as well as what results would refute the hypothesis. Now, the next task in this week's laboratory is to generate and to interpret graphical representations of data. We'll start with the data we gathered last week, the volume accuracy results, and the human measurements data set. As we're thinking about graphing, one issue that stands out front and center is the determination of which variable is independent and which is dependent. Identifying which is important, especially now, because you almost always want to have the independent variable on the horizontal axis. Graphs are used to visually depict a pattern or an absence of a pattern in the data. By looking at a graph with a horizontal and a vertical axis, you should be able to say, first of all, whether or not there is a pattern or relationship between the two variables. If there is a pattern evident, what is it? And what does it suggest? A lot of this week's activities will require collaboration with the other members in your work group. Ideally, everyone in your group will have done their part in diligently preparing for the laboratory, having completed this video and having read through the written instructions of the laboratory carefully. Even under such ideal conditions, there will likely be some differences in perspective between the members of your group. If you think one way about a particular answer and other people in your group have different ideas from yours, you should discuss. Is it possible for different answers to be correct? Why, yes. When it comes to experimental design, there are usually several different ways to approach any given question, and it's a good thing to see other ideas and to consider the merits they might have. Given this kind of flexibility, is it even possible to make a bad suggestion? Oh yeah, there are many, many more ways to be wrong than right. And if there's a bad idea being aired in your group, you don't need to shout anyone down, but the misguided person needs to be redirected. And in the context of a group discussion, that responsibility falls to those members of the group who can explain where the faults are in that misguided person's thinking. You should also remain open to the possibility that this person is actually the one with the correct picture. If they can validate their thinking with sound reasoning, it might be that you end up deciding that you were the one in need of some redirection. Now, if all the questions I gave you were going to be easy and with one obvious answer, there would be no need for group discussion. Don't expect a lot of those easy questions in this lab class. The greater the challenge, the more the group needs to work together with substantive contributions from all of its members. By the way, don't get too attached to your lab partner or your lab group. I'll be reassigning students to new groups every few weeks. Over the semester, you'll be working closely with most everybody that we have in the class. The last thing we do this week is to work through an entire experimental sequence, starting with an observation which is provided to you, and from which you and your group move through all of the steps of experimental design, the actual experimentation, and ending up with graphical representations of your results and conclusions. As always, everything we do should be recorded in your lab notes.